University of Burgundy.
Yes, there we go. So with that,
Oh, okay. Thank you very much for the invitation and finding time to join me. And so today I will talk to you about computational biomarkers with imaging and alternative data sources. This is going to be a journey where I will explore some of my research and try to show you the link between all of it and how it can be seen as ways of creating different computational biomarkers for health applications. So before I start, I'd like to make clear what I intend personally for computational biomarker, and I'll start by saying what is a biomarker according to FDA and NIH. Essentially, it's a measurement of normal pathogenical biological processes or response to therapy. This can be used for diagnosis or treatment response or screening or potentially other applications. We can calculate biomarkers from lots of different sources. So from the very small scale, like DNA, RNA, molecules, then going up protein cells, but as well we can calculate biomarkers from larger structures, so large body structures like the one we see in MRI imaging or physiology itself. And generally, the more we go to a larger scale, the harder it is, because there are multiple factors that play a role. So what is a computational biomarker? The way I see it is that a biomarker that takes multivariate data, so a lot of variables, and then extract the essence of a single measurement that is relevant to measure a normal or pathogenic biological response or response to therapy. So here I put a little funnel. Obviously, it's not the whole multivariate data that we squeeze into a single measurement. It's really a machine that goes here and extracts the relevant information for obtaining this biomarker. So with that squared out, let's go to the central representation. So it will be divided in three parts. The first part, I'll show you biomarkers to create diabetic retinopathy screening with retinal fundus images. That's the work done for my Ph.D. at Oak Ridge, University of Burgundy, and as well with the University of Tennessee. Then the second one, we use videos of mice, of small animal behavior, and the idea is getting high throughput and social behavior analysis biomarkers. And the third one, which is my most recent work at MIT, is using the interaction with digital devices to get early diagnosis of Parkinson's disease. So all of these are examples of biomarkers, and they follow a pattern that is common for machine learning research, but is increasingly common also on other types of fields. So what happens is that we have the raw data, our signal. Then we have low-dimensional representations because, as we said before, these computational biomarkers have to take a large of multivariate data that in some aspect they can be seen as dimensions. So when I talk multivariate or dimensionality or features, I'm talking really about the same thing. So for one sample, you have loads of numbers. Then after representing it with lower space, we have a system, a machinery, which can be an automatic classifier, regressors, or you name it, that distills a numerical score. This is very abstract now, and then I'll try to use the schema later on to explain really how it's applied for the different type of techniques. Aspects that are very, very important are two. First, the training. So these classifiers need to learn generalizable patterns. This requires a data set plus a ground truth. The ground truth is between inverted commas because oftentimes it's not really ground truth. So, but it's our target, something that we want to learn to generalize and get in this our numerical score. This can be a diagnosis, an endophenotype, or domain knowledge in general. Now, other very important aspect is the validation process. Once we get the numerical scores before we can really call it a computational biomarkers, we need to test things like repeatability, specificity, sensitivity, which is very important. We don't want something that works on everything because nothing works on everything. Now, let's go to the first one. So, retina image analysis. I'd like to start with first moving this thing out of the way. Yeah, like that. I'd like to start each project, each branch of these three computational biomarkers, with an unmet medical need. So what drove really the research? In the first case is essentially blindness and diabetes. We know that there are 135 million diabetic patients worldwide. This is projected to increase substantially. And 1/3 have retina complications. These are generally diabetic retinopathy or macular edema, which are very, very intertwined. So this is actually the primary cause of blindness in adults in the world, because there are a lot of diabetic patients here. And why is it so problematic? It's because people do not realize that they have it. What happens is that you have lesions on your retina, and essentially you are blind on a very, very small spot, and your brain fills up that spot because our brain is really good at filling up this thing, are generalizing data in general, what we see. And so you don't realize that you are losing your sight slowly. So even when you have mild diabetic retinopathy, you have a perfect vision. You can have 20/20 visions. But then when it gets severe, you'll have essentially black spots all over your eye, and then you'll become eventually blind. What is the good news? The good news is that if you detect the disease early, you can treat it very, very easily and cheaply. Essentially, they will burn part of your external part of the retina, which will allow the vasculature not to grow in there. They require less oxygen, and so that you will not go for the bad effects of the diabetic retinopathy. The problem, short of ophthalmologists, you have to get an eye exams to see this thing, which is relatively easy to do, but there are not enough ophthalmologists to check all the people, because if you are a diabetic patient, you have to do a check every 6 months to make sure. This is what's suggested. So what is the opportunity? Create image-based computational biomarkers for retina screening. Use as source image the fundus images. The other good news is that fundus imaging is very easy to achieve. This is a machine that looks a bit scary, but in reality is nothing more than a camera that shines some light into your eye, and then the back of the retina reflects the light, and this is what you see. You see this type of images that they do not require actually even pupil dilation, so you don't have to put any drops or anything like that. Some of the structure of the retina. This is the optic nerve. This is where all the vessels and the nerves go from the eye to your brain. This is the macular region. Is the area where the highest concentrations of cells that allows the vision are in. So if you lose this part, it's very, very bad. If you lose the part around this, is not that bad. And these are examples of some different type of lesions. We have these are exudates. These are some types of hemorrhages. And here I'm showing other examples of the retina to show what are the main challenges of this problem. The retina is very, very different in each one of us. It's very unique. Depending, for example, on the skin color, will change a lot. These are people with darker skin tones. These are people with lighter skin tones. There are things that are normal in the physiology. For example, this person here has all these stripes that looks a bit strange, but is perfectly normal. This person has all this white part, which is normal as well. This person instead has lesions and these white things. So these are bad stuff that we want to detect. These are not. Now, let's go more into the lesions that we want to detect. The lesions we want to detect are mainly microaneurysms, which are little inflated capillaries that start at a very early stage of the disease, and then exudates, which is essentially lipids that leak out of the microaneurysms, and they create residuals, like something that you would get into your sink. Then, this is the very early stage of the disease. If it not gets treated, this is what will happen. Essentially, the microaneurysms will leak blood, all of this stuff here, and they will create new vasculature, and all sorts of bad things happen, and you become slowly blind. So now, I'll start my talk to show a technique to create a low-dimensional representations for detecting microaneurysms. So going back to our model we showed before, our raw data will be little windows of the retina images. So for windows, I mean little subparts of the retina. And then we'll create a low-dimensional representations with what was a new thing that I developed during my PhD, which is a radon descriptor. And then we'll use a standard automatic classifier regressors, and which will give us a microaneurysm score. I will concentrate on this one only for this part of the talk, to give a sense of how you could represent with low dimensions based on domain knowledge. So, this is a model of a microaneurysms. It looks pretty much like a Gaussian, very no noise. And this is the representation in the radon space. So what is the radon space? This is a transform that is used a lot in MRI, or pardon me, in computer tomography to do 3D reconstruction. In here we are using it for a completely different purpose. So in the image process, we want to detect good measurements in the low dimensional space, that particular microaneurysm. So let's go over how these things are calculated. So the thing is, we get these rays that go through the image, and then we integrate the value across these rays. This is, for example, the first one, and we'll put it here. Then we'll integrate the values across this ray, and we'll put it here. So for this projection, you will get a single line of this graph. Then we rotate this stream of rays, and we'll project it in here, and we'll do it again for the next number of rotations. As you can see, this is a Gaussian, and it has this nice ridge-like shape of a mountain on the Gaussian. So I say, well, it's fine, you know. I could have done that with by fitting a normal Gaussian to it. It would be fine. Well, it turns out this has an interesting property. When we add a lot of noise into it, still we see this mountain-like shape with a crest at the center. And this again, yeah, well, even with some Gaussian model, some trick here and there, probably could have get something similar. But what about if there is another long structure that is very prominent in the window? In that, you would not be able to fit any type of Gaussian model. But still, in the sinogram, you'll be able to see a crest that is continuous in here, this ridge. You will still see a peak in here that represents the vessel, but will be discontinuous. Instead, there will be a continuous part in the center of the space that represent these little lesions. So we want to exploit that to create a space representation of this image. So these are examples. They look very bad. Example of real microaneurysms with the vessels close to each other. They are already low quality even here, but this is even worse. And this is the representation in the radon space. So what we did, we take other rays in here, and we calculate the mean across this direction, which creates a vector of 19 pixels, assuming a window size of 17 by 17. And then we take the standard deviation, and we put that into a feature vector. More precisely, we take the derivative of the mean and the standard deviation itself of size 37. So we went from a dimension of 17 by 17, which is a little bit less than 400, to 30 to 37. And we'll put it here. Now, we could start already to run our classifiers and getting our numerical score, but actually there is something missing. It's the data set. The problem in here, the challenge, is that the data availability is not excellent. While it's easy for an ophthalmologist to give us examples of lesions, it's very hard to find examples of true negatives. Why? Because the image is it's made of tons and tons and tons of other things, and asking the ophthalmologist to look at every little part and tells us if it's a lesion or not is simply not possible, because for most of the image, they don't care. There might be something, but they are not really sure. So to challenge this problem, what we did is resort to active learning. So this is an image of the retina. I took away some of the channels to enhance actually a little bit the vessels and the lesions. The lesions are these little things in here in the circle. So you can see that they're very tiny. Now, what we did is starting to train our pipeline with a very simple a few examples taken from a few images, and then we run it. And in here we have examples of the lesion scores. The lesion scores goes from 0 to 1, where 1 is black and 0 is reddish white. The more they tend to black, the more it's likely to be a lesion. The good news is that inside here, inside those windows, we did find the lesions, but on the other hand, we found all sorts of other stuff all over the place. In active learning, this was a real-time system. We asked the expert to click on the areas they knew they were not lesions, and then iteratively we retrain our classifiers with those examples, and then we show live what's going on. So if you're familiar with learning algorithm, it's like if our expert was a gradient descent. So it was looking like and it was moving forward. After adding 5 negative examples, this will happen. After other 5, this will happen. After other 5, this will happen, and we got to here. So this literally took 10 minutes, and for this image, we're getting excellent results. If we run this classifier with just a training on a single image on this one, I'm highlighting all the lesions in there, but also other structuring here to show what's going on. We obtained some really good results. So all the stuff that is outside, they have a low score probability, while the stuff that is inside the windows have a high score probability. So just to reiterate, we get some initial sample, we train the classifier, we generate the scores, evaluate our results on the training set, very important. If there are too many false positive, click on a false positive, we add it to the classifiers, and we go again. And then finally we store the final samples. This is obviously not the whole story, because this is what we did for the microaneurysm. Our final target is creating a retinopathy screening market. So taking the same model we had before, what we do, we add our retina image and the microaneurysm score we obtained, but then we'll go again through a low-dimensional representation of this microaneurysm score, and we train classifiers to detect diabetic retinopathy. I will not go into details how it was done, but essentially we create a space that is regardless of the number of microaneurysms, we can see it, and we can get features that are the same regardless of the image. This is a ROC curve that shows our performance. As a reminder, the ROC curve shows the graph of sensitivity and specificity. So the worst ROC curve we could possibly have is a line that goes from here to there, which means that the classification is by chance. The more you tend to this direction, the better it is. For each point, you get a couple of sensitivity and specificity. For example, in here, we got almost perfect sensitivity and 50% specificity, which is very good for our problem, because we are creating here a screening market. So with a screening marker, what we want to do is make sure that all the people that have the disease are screened, so we detect the disease, and we don't care that much about if we get someone wrong, because the risk of getting someone wrong is much higher of getting someone of losing someone that is good. And using this classifier with 50% specificity, we will reduce by 50% the visit with ophthalmologists. So since most of the people will have nothing, this is a very large number improvement. So this was the curve for healthy versus high risk of developing disease, diabetic retinopathy, and the other one is healthy versus any risk of detecting diabetic retinopathy. This was done with 1,200 images and 800 images on an external data set. And actually the algorithm was trained on nothing on this data set, on another one, so it showed some really good way of translating the data. Now I'll talk very briefly also some results we got on macular edema screening. The pipeline is similar, but the way the classifiers and the low-dimensional representation is different. In here, what we did was using image features. So we did not care that much about the lesions themselves. We did a little bit, but a lot of features that seems really good are image level, but I will not go into the details. What I like to show as well, the comparison, which I think is very important, between our classification strategies and the experts. So essentially, we have a data set. We took a subset of the data set with 350 people, and then we asked the expert one to evaluate those images and the expert to evaluate those images, so we could compare our technique with them. And the good news is that we can get the same sensitivity of the best expert, again with 0.5% of 50% specificity, which is again it's a really good result. And we published that in medical meta-analysis, and we were very happy about that.
Did these experts train your active learning classifier, or was
No.
it trained by Zoom?
No. In here, by the way, there is no, in this particular case, we did not use any active learning. The active learning was used for detecting those the microaneurysm scores.
I see.
So this is a summary of the work done in generally for diabetic retinopathy. We developed these two screen metrics, and we take the full pipeline, executes in 22 seconds. This includes another algorithm for doing the quality detection, so that takes in or out images that are not of good quality enough. There is a commercial spin-off using part of this technology, which was acquired by Welch Allyn last year, and we published, for some conferences, journal, and book chapters. So this concludes the work on retina fundus images, and now we are moving to something that is I find quite exciting. As you can see, the mouse here is pretty excited with his saxophone, and it's with videos. So we want to find computational biomarkers using videos as our data source. What is the problem here? What is our research trying to tackle? Well, behavior analysis is essential to study phenotypes in animal models. What you know much better than I do. You knock out a gene out of animal models, and then you check how, with some tests, what's going on, what is the effect, what is the phenotype, or you test a drug, and you see again with measure what goes on. Most of the experiments so far are limited to single animal. And this is a problem for doing tests to study autism, schizophrenia, and anything that has a social component into it, which is some really exciting areas of research so far. So to do that, we need people that the constant human supervisions, and then the manual scoring, which is hard to get and is low throughput. So what we have developed is a system. It's a vision-based system. This is the schema. So this is a thermal camera. We have our mice in here. They are placed in an arena. So it's an open arena. They can interact as they want. There is no tagging on them. And this is what was one of the constraints the life scientists told us, because if they put tagging on them, their behavior will change. So if they remove some fur, they will do something. If you add some paint, they will smell it. So it was hard. This is the biggest challenge, trying to find the identity between them, because they will also cuddle up together. They will jump on top of each other, which makes it challenging. So this is the result of an example of our tracking algorithm with 3 mice. So the little points are put by the computer vision algorithm we developed, which is runs live, by the way, in real time, and you can see them going around and doing things. So it's really resistant to inversions of the animals. Having said that, it's not perfect. So as well, we developed in the pipeline a system that evaluates the risks of having an inversions, so that the people could evaluate in a very fast way if those animals were exchanging identities without watching the whole video altogether. This is the tracking algorithm was based on a hybrid approach. It was based on the position with an extension of the temporal watershed with a maximization framework, which deals with occlusion, as well as a heat signature. This was one of the key components. So that this is a thermal camera, so we can get very precise measurement of the heat distribution, and the heat distribution stayed more or less constant across time. So we use that to disambiguate between the identity of the mice. These are an example of the path of an experiment where they had wild-type mice, so normal mice, as well as schizophrenia model. And even visually, you see the schizophrenia model going like a maniac, like it was expected.
Can I ask a quick question?
Yeah.
For the schizophrenic mice, does their temperature stay consistent when they're aroused?
I don't know, to be honest to you. Then we went one step forward, and what we really want to get is, okay, this is nice. I mean, the path is something that is typically done with a lot of other systems, but what we want to do is gauge in the social behavior to enable these experiments. And social behavior, the description of behaviors, is very qualitative. So some people start to put rules, say, okay, if they follow each other for less than X, this happens. If they do this, less than whatever. But the rules change slightly for each person. So our idea was to exploit examples. So the expert will look at the videos and will give us examples of this behavior, for example, the nose to body, this behavior, the nose to nose, the following, the stand together, the stand alone, walk alone. Then we will use our pipeline, where we will get as an input the mice relative position. So this is interaction. So it's not the actual absolute position, but it's the relative, and then their shapes across time. Then we'll go to the low-dimensional representation, and we'll train our classifiers with examples, and then we'll get the mice behaviors. This is an example of our classifiers. I will not go into details. Is based on decision forests. I just want to highlight that one of the main challenges here is that the combinatory aspects of behavior. Since it's social, you have if you have 3 mice, you have already the combination of 1 versus 2, 1 versus 3, and 3 versus 2. So this is pretty hard to make. And when it's 4, is even more. Here is an example of the results of the classifier. You have the 4 mice, and in here some of the behavior. And we could, in this way, run very long experiments and look on how long they went to stay on one behavior or on the next. So again, I know a bit more about this experiment. It's because the very first on the wild type, it was really our pilot data. In here, instead we took 10 wild type mice and 6 autism mice models, BTBR. We calculate the way they interact, and we could see that the social behaviors in the wild type mice is much more than the statistically significant more than the BTBR as expected. This was published. We was very happy. But then the people in there at the life science, we used this technique to actually making discoveries, because this is something that it was known is an autism model. So you know that these mice are less sociable. This is what they test. They tested the oxytocin treatment done chronically intranasally, and they discovered that decreased the male-to-male social interaction. This treatment it was a potential candidate for various neurological diseases. It was the first time this was proven, and they saw that with a high dose of this treatment, they could see the social behavior decreasing. But even with a low dose, they could sort of get the difference, although it was not statistically significant. This was published in Neuropsychopharmacology 2 years ago. So some conclusions. We were the first multi-animal behavior by example system. It's allowing new high-throughput life science experiment. We started industrial collaboration with Techniplus, which are producers of cages. We got 2 international journals and 2 peer-reviewed conference. Now I will. By the way, this is the work that I've been doing at the Italian Institute of Technology during my postdoc. Now I'll move about my work that I've been doing at MIT for the past almost 3 years. And in this case, we'll use as source a different type of data, something that we believe has not been used or explored enough before. And the aim will be the diagnosis of early Parkinson's disease. Let's start with the unmet medical need. Parkinson's disease affects 5 million people worldwide. The prevalence is expected to double by 2040. Something that you might not know is that Parkinson's disease does not affect the elderly only. It's 20% of the patients are diagnosed under 50. But this type of neurological disease is very subtle, so the decrease is very subtle. So most of the people have Parkinson's, they don't tell anything, and they just go on with their daily life. We know that prompt diagnosis improves the outcomes, although there is not a definite cure, and enables two therapies. This second part is very, very important. Why? Because in Parkinson's disease, once it's diagnosed, it's really potentially too late for having a cure to stop the dopaminergic brain cells death. If the clinical diagnosis is possible after 10 years of the disease onset, because there are the first perceivable symptoms, so you can diagnose it clinically. However, there are some studies that shows that there is a subtle motor impairment that starts much earlier where the clinical diagnosis is possible. So we want to exploit that to push these clinical diagnoses as early as possible to enable new neuroprotective treatment, but as well finding a way of improving the outcome for patients that have been diagnosing here. The best measurements that we have so far they are called the UPDRS score. They are the most used motor scores, and it involves the patient going to a neurologist, to a motor expert. Actually, not all the neurologists can do this type of tests. And then they'll ask the patients to extend their finger like that to measure rigidity. Then they'll measure their balance. They'll ask them to stamp their foot, looking at gait, look at rigidity again, the tremors by doing that, and some other tests. They give for each. These are not the whole story. There are all others. And for each test, they give a score from 0 to 4. It's summed together, and this is like the best scores we have to evaluate Parkinson's. This is qualitatively, it's good, it's very good. But once you want to get longitudinal change that are very subtle, this does not work very well, especially at the early stage when the scores are very tiny. So we want to create a new paradigm with the high compliance and frequency test that does not require the doctor, so you could get a lot of measurement longitudinally, and integrates with daily activity, so we do not create a burden for the subject. As well, again, we want to do at-home data capturing to get this change about the daily aspect of the disease. And we want to do that with a technique that is at our fingertips. So essentially, the interaction with the digital devices, it might be a tablet, a laptop, or a phone. So what we are discovering is that by judging how each finger hits a key or a screen can be good to infer a motor score. That then can be used, this motor score, for potentially early Parkinson detection, tracking, and other potential applications. What are we measuring? What we are measuring is essentially the time it takes to press and release a key. This time we know from studies done on keyboards for completely different reasons, because they wanted to measure how good the keyboards were. It's around 100 milliseconds. So you can imagine that this effect, this measurement is very subcortical. So it's something that you cannot really change it if you want to. I mean, obviously you can go 1 second hitting the key, but the subtle changes in this timing will be very hard to do it by yourself. So we used this measurement, and then we took again our pipeline, and in this case our input data will be the key hold time series, so the timing of this key and the progression, and then we'll go through that, and we want to get out of it a Parkinson's disease computational biomarker. As you can see, I mean, this idea I presented can be a little bit far-fetched, so there are loads of questions that can pop up into the mind. What about if I'm a skilled typer? What about the text I'm typing? What about if I have a coffee? So, no, what we did is was to start with somewhere. We want to start with some ideas. Well, first of all, we looked at medical imaging. So this is a study, a review of 16 PET, functional PET, and 22 functional MRI studies that looked actually at finger tapping. So it's not really tapping, but it's literally tapping a finger, and this is typically done to study motor effect. And we saw that the networks that gets activated are compatible with Parkinson's. So this was already a good start. Then we decided to run a pilot study. The pilot study at MIT, obviously, we had at the beginning we had limited resources to prove this idea. So what we did is use as a proxy something called sleep inertia. Sleep inertia is what happens when you are getting up in the middle of the night, and is an effect that is studied. It creates a motor impairment that is comparable of being sleep deprived 24 hours, more or less. So what we did is taking 14 MIT students in exchange of a Chipotle card, and and then we asked them, we installed on their computer a plugin that was running on Chrome that could capture this type of data, and then they asked them to type a Wikipedia page of their own choice during the day. Then we were giving us a call during before going to sleep, and we were waking them up in the middle of the night and asked them to type again. By doing that, and the text was different. By doing that, we were able to get initial data to test our hypothesis. I will show what the results are, but I want also to show what is the technology development timeline. After this, we've proven that, I'll give you like a bit glimpse into the future representation. We proved that we could do it. So then we moved into testing late-stage patients. Then we moved to recently diagnosed patients, and then a study on the very, very first diagnosis. So we are going towards this direction of the Parkinson's disease progression. But let's concentrate a second with the pilot study with the sleep inertia. As I told you, this is a state of grogginess comparable to being sleep deprived for 14 hours. The effect lasts from 15 to 30 minutes. Our pipeline, in this case, is getting a motor impairment score, so not a Parkinson score. 14 subjects, we did the repetition twice, so we had four data points. People had different languages, different laptops. The data was collected at home, and the typing task was for around 15 minutes. The results were published on scientific reports last year. So, what low dimensional representation we used? This is very, very different because the keyhole time series have not been studied a lot, and it's challenging because it's very sparse, the data. You can imagine that while you are typing, you have large chunk of moments where you're doing nothing, and then you type some burst, and then you have some other burst, and we had to find a way of representing this signal. So what we did is taking the signal. This is the typing time. This is the actual hold time in the order of milliseconds, and then we create some sliding window. For each sliding window, we compute the normalized distribution with the hold time histogram, and then we take these histograms, and for each one, we'll put it in a matrix like that. This will create a sort of spectrogram-like representations, which then we can analyze in different ways. Now I'll show you some examples of these spectrograms. They're really cool because by just looking at them, you can sort of see what's going on. On the left, we have the arrested patients. On the right, the patients with, the subjects, they were healthy. There was nothing wrong with them. The subjects with sleep inertia. And then I put in order the difficulty of seeing an effect. So on the top row, we see even visually a strong effect. So what we see here is that essentially the mean of the hold time probability is lower. So it goes from this is 200 milliseconds, this is 0 millisecond. It's lower when you're rested, and instead is higher when you have sleep inertia. But more importantly, there's also another effect that the variance of each little column, it changes, and not just the variance, the distribution carries on changing a little bit. In the second one, we don't see that much the change into length of hold time, but we still see a lot of change of this change of distribution. This is an effect that essentially violates this heteroscedasticity in a statistical term, a change of variance every time. The third one is a little bit harder to see with the eyes, but with our numerical representation, we see it very, very well. So this is our numerical representation. I won't deal too much into details, but essentially there are two features. One is the average of the column media, and the other is a measure of connectivity, where connectivity is measured with a matrix that measures the distance between all the different columns, so all the relationships. So we build essential network across all of it. If we plot the two features with the absolute value, we have in red the sleep inertia and in blue the fully awakened. We sort of see something, but yeah, we're not very excited about that. On the other hand, if we take the delta between rested and sleep inertia, this is what happens. Pardon me, rested and sleep inertia, and then sleep inertia versus sleep inertia, and rested. We see a very, very nice distinctions between the red, which is from awake, the delta from awaken to sleep inertia, and the blue, which is the delta in similar state. So both, let's call them asleep, and both awaken. They are statistical significant, but still we train the classifier with hold out approach, and to get actually a motor impairment score, so a single number that could represent these two things. Here are some ROC curve, and we compare on the red what we achieved with the blue, which is just the whole time, so the median of the whole time, and with the green, which is the average typing speed. So we show that in here our metrics is much better than your own typing speed, the difference in your own typing speed, which I would have thought that actually that would have been quite different, but actually is not. So this is all for the pilot study on sleep inertia. So we start and say, good, we have something, let's try it on Parkinson's patients. We had this patient 0, which is a late stage Parkinson's disease patient, and this patient has a deep brain stimulator. This was really good for us to judge the difference between symptoms, because the patient can turn on or off. When it's off, we'll have more symptoms. When it's on, we'll have less symptoms. And we were seeing somehow a sort of similar effect visually. So you see that this thing, the variance increases and it goes down. But we were also seeing a new effect. These new effects were in there regardless of the deep brain stimulators, which were these outliers in there, which we thought it could be connected to dyskinesia. Dyskinesia are involuntary, not voluntary movement. The thing is that this is a late stage patient, so we did not want to base our study on a late stage patients population. So we went for the harder one, to early stage population. So we got patients that definition of early stage that have less than 5 years from diagnosis and have minimal or no functional impairment. Essentially, it's very hard to tell from an untrained eye that they have Parkinson's disease. It was a controlled population in the sense that we matched by age, sex, and the skills of computing skills. 13 controls and 18 Parkinson's disease patients, and we keep them without medications for 18 hours. The tasks were a 15-minute task of typing in the clinic. Then there was a clinical evaluation with PDRS, and then another task that is done a lot in clinical trials for neurodegenerative disorders, which is finger tapping. And we had 2 repetitions of it. The aim was to create a quantitative scores to actually separate the classes with their absolute values, something that we could not get with the first experiment. The first thing we tried, actually, it was running exactly the same features, and actually it didn't work. It didn't work, so we had to go back to the drawing board and think about another type of technique. So we took a similar matrix representation. We added windows that were larger, so each column is around 90 seconds ahead this time, and then we added other features that evaluate variance. So this is to capture more of these outliers. So here we have features that are related to quantiles, to the number of outliers, to interquantiles, quantiles range. And then we'll adopt an approach that actually try to learn the patterns from the data. This is because this time we want to create a system that is much more generalizable to other type of patterns. And to do that, first of all, we checked if little column was looking like with an unsupervised algorithm that probably most of you know, which is the principal component analysis. We considered each 90-second sample as an full independent samples. So we had 570 samples from those 18 Parkinson's disease and 13 controls. And what was really nice that even with this simple linear projection on 2 principal components, we could see that most of the time people were standing here, but sometimes there were these effects. They were getting out of the main mode of the distribution, And these were only the Parkinsons doing that. So again, each little point is worth 90 seconds of typing. So we interpret that as instances in which the Parkinson's patient get Parkinsonian effect, and we base these algorithm on this type of initial analysis. So even if project on the first postdoctral component, you can see the distribution, and you can see this nice part on the tail in here. So I don't want to go too much into details again about the classifier and the regressors, but essentially what happens is the classifier uses these 90-second samples, and then it runs with a bagging approach loads of different little estimations. Each little estimations has a sort of manifold learning, which is based on linear PCA, and then we have a regression component based on supervector machines with a kernel. We do that by looking at the data sets in different way. We get a lot of scores, we aggregate them, and then we get one single scores from these 90-second samples. Very importantly, every time we do one of these iterations, we make sure that at least a few patients are left out so that we could use them as for test set. So we do not absolutely want to train this part of stuff and then test it with the same data, which is very challenging to do with a data set that is that tiny. What we got? So these are the scores for the joint population. We have on the top the median, which is this line and this line. This is the upper quartile, lower quartile, upper quartile, and lower quartile. There is no smoothing whatsoever done into this thing. This is like literally the pure data. We take the first 90-second window, and then we get the median and the quartiles for Parkinson's and for controls. And we do that for the other windows. So even consider them independently, we see that they are well separated. And this type of representation is quite interesting to compare it with actually the typing speed in this window. The typing speed is very bad at separating that. So they are very much on the same distribution. And also it's important to say that in our test we had some patients that were touch typists and that were single finger typers. So now this is the analysis, the ROC analysis for these patients. We have in blue our score, and in green we have the comparison of the alternated finger tapping, which is the test that they use for clinical studies, which involve the following test. You take two buttons, a machine with two buttons, and then you go like that for 60 seconds as fast as possible, and then you get the metric. And then this is another type of finger tapping, which is used again a lot in clinical studies, which is that involves clicking as much as fast as possible a single key, which instead was pretty bad at distinguishing our two classes. So this is under review on scientific reports as well. Now the data set that we use, we think we controlled everything, but obviously it's quite small. So what we did, we started what we call a crowdsource effort. So we have a website, and we ask people to fill out the form with their age, sex, and their condition, and then to ask them to download the software to install on their computer, and then we'll monitor what they were doing. Very important. What we do is measuring just the timing. We do not get any type of keys. So what they are pressing, we don't know. Not even in the algorithms you saw before. The only thing we do is taking away the keys that you are supposed to press for a long time, like the shifts, controls, and this other type of things. This allows us really to look at the effect, the type of characterize the typing in the general population. So this is the data we have the 63 controls in this uncontrolled study, and each one of them typed on average for 55 hours. The challenge here is that the task is very, very different, because in here they could do whatever they want. Instead, in the clinics, we knew that they were typing a text. The text was different for each one of us to avoid any learning effect, but still, we knew that more or less it was a continuous typing effect. So if we add these 63 controls in our data set, we are very happy, because the performance go down as we would have imagined, but not that much. So that means that this technique could be, with some adjustment, could be used onto this population, and we could potentially use that for tracking at home during the day. This is a comparison, by the way, to give a sense of how good the performance are of the clinician's performance, of not actually clinician, of neurologist performance of diagnosis Parkinson's in general, not even early. This was a study done on a different population, so take it with a pinch of salt, but is to give an idea of where we stand. So we then, after with these results, we went to the Michael J. Fox Foundations, which are some thought leader of Parkinson's disease, and we decided to create another study, this time monitoring the Parkinson's patients at home as well in the clinics. And instead, we'll get them as the very, very first diagnosis. When they have taken no drugs whatsoever, they are at the very edge of the clinical diagnosis. And not only that, we want to follow their treatment. So the idea there is to say, look, we show that we can get a motor disease score change with the pilot study. We show that we can detect the two families of Parkinson's versus controls. Now we want to show how good we are at detecting a intra-individual change, which in my mind it should be easier, because when you are your own control, it's the effect of the typing and what you do and the way you behave. Are bound to change much less. So this study is ongoing, but I want to show some of the very, very initial results. So take them as with a pinch of salt, but these are real patients. This is real data. So in here, we have plotted what's happening with these Parkinson patients. You have on the x-axis the weeks of the follow-up, and in blue you have our neuroQWERTY index, which is exactly what was used on the other data set. There is no change in a training. There is nothing. We want to keep it as clean as possible to see really what's going on before we start with messing around with the data. And in black you have the clinical evaluations, which is the UPDRS scores. And the more both things go down, the less symptoms they have. So the more up, the worse Parkinson's effect they have. They go down, the better they get. Each point that you see in this point and this point and this point are the 3 visits they got. And then in the neuroQWERTY scores, each point is simply the average of 3 days. Again, there is no weird modeling going on. It's literally taking those 90-second samples, getting the score, and average with the brute force way the algorithm. So there is much more that can be done. But it's nice because we do follow the same trend. And if you look at when the medication started, it's consistent with our finding. And also, we do see a change in here. Well, obviously we did all the statistics and everything, but in here we see much earlier of what's going on for the UPDRS study and for the clinical visits. And I mean, it's quite understandable because we have our method that run passively and it's done at home, while the people can go to the doctor with much less frequency. This is an example of a skill-matched control where instead it's pretty flat in all locations. So now, I mean, we are running a study. Recruitment will be finished next month. So hopefully in a few months we'll be able to get actual statistics and nice analysis out of it. So some conclusions of this part of the talk. We show that passive interaction with digital devices has a physiological significance, which was not shown by anyone before. We developed actually the technology to do data acquisition with the multicentric environments. We have all our servers, our clients that run on multiple platforms, all the IRBs and everything. We developed 2 pilot metrics, one of sleep inertia and one for early stages of PD. Something also that I forgot to mention is that when we try this technique, this metric on the sleep inertia data sets, all the sleep inertia people are classified as controls. So they are down there in the metric. They are down here, which is very good. Then we started a collaboration with Michael J. Fox Foundation. We are starting now a collaboration with a pharma company that the contract should be signed by them, hopefully in a few weeks. We won a prize in Singapore. We published in an international journal, and then we have other 3 under reviews. So this concludes generally this journey across computational biomarkers, which I hope I kept you awake. I know that after eating it can be very challenging. And I want to conclude with some quick ideas for the future, and what in particular about precision medicine. So what's next? So that we know that precision medicine, the main aim is to move beyond the one treatment fits all model. And I think that can be accomplished by providing a complete and actionable information to clinicians, researchers, and medical practitioners in general. And I believe that the computational biomarkers will play an important role in that. I see that as creating a sort of batch of these computational biomarkers, and that can include information with genetic, imaging, medical history, and generally health techniques. By doing that, it opens all sorts of possibilities. Let's say that you go to your eye doctor to check if your eye is fine. You get that image, and then you can run in the background all of other batch of computational biomarkers. You could see the risk of other diseases, for example. Or you could find potentially some relationship between imaging and genetics by correlating the biomarkers you get, the computational biomarkers you get into two. Or you could even use the same technique by using the data from one and the other and get a joint biomarker that's used on both of them. This, however, it can be accomplished only if we move beyond the custom-built solutions. So we do not want to build every time a different technique. We want to create a model that it's ready to apply for different problems, and is entirely based on the data. This is becoming now possible thanks to the amount of data that we get, and the ground truth, and the new techniques like deep learning, supervised, semi-supervised learning, that starts to learn a pattern on the data itself. And then we can apply the standard classification techniques we are doing for that. So learn the models from the data. So a lot of acknowledgements. So I put people, and probably I'm forgetting a lot of them. As you saw, I'm a little bit of a gypsy. I've been moved around a lot, but I would like to thank all of them, and thank you in particular for the invitation. Let me know if you have any questions.